Eosinophils were once known only for their role in allergy and parasitic infection. Today, we're uncovering their far-reaching influence in immunity, inflammation, and beyond.
Eosinophils are specialized white cell. While they participate in immune responses, they are also key players in allergy and asthma and in other chronic inflammatory diseases. New data suggests eosinophils may also play pro- and inhibitory roles in many other areas, including tumor development and neural growth.
Understanding eosinophil biology is essential, not just for treating these conditions, but for advancing our overall understanding of the immune system itself in both health and disease. That's where the International Eosinophil Society steps in.
IES is a unique platform where clinical research meets basic science. Our members are driving discoveries that move from the lab bench to the patient's bedside.
What sets our society apart is our seamless integration of disciplines. By bridging clinical practice and foundational research, we unlock new possibilities. IES is more than just a meeting. We're a society of individuals of all parts of the globe, interested in using science to best understand the eosinophils and rapidly apply that to the benefit of patients with eosinophilic diseases.
IES has provided me with invaluable opportunities to connect, learn, and grow. Through the society, I've met mentors, collaborators, peers who have profoundly shaped my career. It opened doors I didn't even imagine, allowed me to share my research globally and find mentors beyond my university who truly believed in me.
IES values collaboration, mentorship, and different perspectives. We bring together people from around the world with different backgrounds and scientific expertise. What sets IES apart is the generosity of the research community. We share reagents, experimental models, ideas, and support to advance science through trust and teamwork.
I didn't realize how valuable this network was until I attended my first IES meeting. It gave me access to science I hadn't seen before and the chance to show my own work with experts I never thought I'd meet.
As an early career member, IES has helped me grow both professionally and personally. It's a community where I felt supported, challenged, and inspired by the work and the people around me.
At IES, we're not just advancing science. We're building a future where eosinophilic diseases are better understood, better diagnosed, and better treated. If you're passionate about eosinophils in the lab, in the clinic, or anywhere in between, the IES is your community. Join us and build that future.
Hello, everyone. I'm Marc Rothenberg, the past president of the International Eosinophil Society, and congratulations. That was an amazing video and truly reflects the diversity of activities and impact that the International Eosinophil Society is having in so many different ways around the world. Along with that excitement, I'm very pleased to have the moderation be a moderator today for the webinar entitled Frontiers in Eosinophilic Esophagitis. Just a little bit about the society, as reviewed in the video. We are an organization of scientists, clinicians who are interested in the eosinophil, a cell strongly associated with many diseases. The society sponsors biennial meetings, monthly webinars such as this, and other educational programs for its more than 300 members in order to review new information about the eosinophil and its role in health and disease. Please join the society. We have some really great promotional offers. Please take advantage of those. The membership fee is currently reduced for regular members by 50%. There's also a one-year membership running from January 1st to 20 through the year, and encourage you to do any of the forms of membership, taking advantage of our benefits, including for trainees, which is free membership for those individuals. And it's open to all people from around the world at all levels of training, including pre-doctorate individuals as those participating in postdoctorate fellowship, clinical fellowships, or residency programs. Please take advantage of these promotions. Also, to mention that membership to the International Eosinophil Society, it comes with free membership to the European Mast Cell Basophil Society. So that's certainly an extra benefit. Want to let you know that we're very excited to already have deep plans in way for the 14th Biennial Congress of our Society, which will take place in Vancouver, Canada on July 12th to 16th next year. It's a beautiful venue and the program is looking extraordinary. Encourage you to visit the website to get more information about that. Thank you particularly to our sponsor today for the webinar, Amgen and AstraZeneca, as well as sponsorship by our Corporate Advisory Council. Members to this Corporate Advisory Council is indicated on that slide, which we'll come back to later on. In terms of a little bookkeeping for today, use the question and answer button, ask questions anytime during each presentation. The moderators will read the questions to the speakers, so you just have to type in the question. All attendees are muted and the chat feature is disabled. The webinar is being recorded and will be posted on the IES website very soon after the webinar today. Viewed closed captions is available, a live transcription during the webinar by clicking on the Show Captions button. So I'd like to now hand over the rest of this introduction to the co-moderator today, Dr. Alfred Doyle. Alfred?
Thank you, Marc. I'm Alfred Doyle, Assistant Professor of Medicine at Mayo Clinic, Arizona, and I'm pleased to help moderate today's IES webinar, Frontiers in Eosinophilic Esophagitis, or EoE. We have an outstanding lineup of speakers today. Dr. Jensen will begin with the epidemiology of EoE. Dr. Nurit Azouz will then discuss the pathophysiology of EoE. And finally, Dr. Evan Dellon will discuss new frontiers in EoE and what the future may hold. As Marc mentioned, please submit questions during each presentation using the Q&A button. We will have a dedicated Q&A period after each talk, time permitting. With that, I think we can go to our first speaker. Our first speaker is Dr. Elizabeth Jensen, who will present the epidemiology of EoE. How much do we really know? Dr. Jensen is a professor of epidemiology at Wake Forest University School of Medicine. She earned her MPH and PhD from the University of North Carolina at Chapel Hill and completed postdoctoral training at the National Institute of Environmental Health Sciences. Her research focuses on pediatric immune-mediated diseases with particular emphasis on the environmental and genetic determinants of EoE. She is an investigator with the Consortium for Eosinophilic Gastrointestinal Disease Researchers, or SEGR, where she contributes to several studies advancing our understanding of EoE epidemiology and treatment outcomes. It is my pleasure to welcome Dr. Jensen. Dr. Jensen, I'll turn it over to you.
Thank you for that wonderful introduction. I just want to take a moment and express my sincere appreciation to the organizers for inviting me to present today. It's truly an honor. So today I've been tasked with providing a brief presentation on the epidemiology of EoE. I'll dive into this in just a moment, but as my title perhaps foreshadows, we may not know quite as much as we might like about the epidemiology of EoE. And by the end of my talk today, I'm hopeful you will appreciate why this is the case. These are my disclosures, none of which have bearing on today's talk. So I'm going to start today with giving a bit of information on what we know about the incidence and prevalence of EoE and how this may be changing over time. But I'll then provide some evidence as to why our understanding of incidence and prevalence may be incomplete given the high potential for undiagnosed cases. And I'll end with just a few slides on the demographic distribution for EoE. Okay, so first, why might understanding of EoE incidence and prevalence be a challenge to really know? So the criteria for diagnosis has shifted across time. And actually, the very first consensus criteria for diagnosis was only published in 2007. And from there have been 2 additional updates, with the most recent guidelines published in 2017 that no longer distinguish PPI responsive disease as a distinct entity. Another challenge is that until recently, EoE was considered a rare disease. So many physicians and healthcare providers were unfamiliar with the disease, and EoE wasn't at the top of the list of possible diagnoses to be considering, especially perhaps in our community-based non-academic practice settings. And then finally, diagnosis requires an upper endoscopy procedure and biopsy. So this leads to delays in diagnosis, especially for those that have either physical or financial challenges in accessing care. And there are missed opportunities for diagnosis, such as when we fail to biopsy a patient presenting with an esophageal food impaction. And I'll provide some data on that in just a few minutes. Okay, but what do we know? So this is the most recent prevalence paper out of the US. And here we examine 2 different data sources: claims data provided through MarketScan commercial claims data, and then Medicare data for claims data representative of older adults. And here, using the standardized prevalence estimates from these sources, we estimated that there are nearly 500,000 cases of EoE in the US, a 5-fold increase in prevalence estimates from just 2 decades ago. But as you are likely aware, prevalence represents both a mix of existing and new cases. So understandably, given this is a non-fatal chronic disease, prevalence will continue to increase with new cases documented. So what about incidence? So this increase in incidence, as being illustrated here in the figure on the left, is 25-fold. This was a little bit of an older study from Denmark, but the nice feature of this study is that they also looked at what was the increase in biopsy rate. So often we hear, oh, maybe it's just increased ascertainment. Well, this study really illustrated that while biopsies were increasing, the rate of increase in incidence far surpassed the rate of increase in biopsies. And on the right, looking at a more recent study in Denmark, we see that actually this is increased an additional doubling in the last 5 years from the publication from ending in 2012. But is there a possibility that the rate of increase may be slowing a bit? So this is unpublished data from an ongoing collaboration with Aarhus University. And here we see that the absolute number of new cases does indeed continue to increase across time, with only minor indication that this may be slowing just a little bit. And in this data from the Netherlands, we observed the same rapid increase in incidence that's outpacing endoscopies with biopsies that I showed from Denmark, but perhaps with more indication of some slowing in this rate of increase. In this next figure from Olmsted County Registry data, and focused specifically on pediatric cases alone, we see evidence of a similar rapid increase in incidence, but with a leveling off or even slight decline from the period 2010 to 2015. And then similarly from this study using Intermountain Health data in Utah, and again in pediatric cases only, we see an increase in incidence initially, as denoted on this yellow line here, in the first 2 years of follow-up from 18.6 cases to 24.8 cases per 100,000, but then a leveling of incidence across the last 3 years of study surveillance. So what are some possibilities of what might be happening here? So one possibility would be that this all reflects true incident cases and that there is or was some environmental factor that led to a rapid surge in cases, but that this exposure has reached its own limitation of proportion exposed, with static new cases, or possibly this exposure may even be diminishing across time slightly, as evidenced in our pediatric data. Another possibility is that an environmental factor contributed to the increased cases, but some of these cases really represented cases that were only newly diagnosed and not newly affected or newly symptomatic. So that some of the surge was attributable to catching up in our cases as recognition of the disease has increased across time. So if this is the case, we might expect that the presentation of those diagnosed would also shift across time. And that's because EoE is known to be a progressive and fibrostenotic disease. So with long-standing disease, risk for fibrosis, narrowing, and strictures increases. And this study, published just last year, illustrated this nicely, essentially showing how diagnostic delay or longer duration of symptoms were associated with increasing risk for a more fibrostenotic presentation at endoscopy. So at the heart of all of this is a question about underdiagnosis. And so is there evidence of possible underdiagnosis? So here, again, examining our cases from Denmark, we see that the median age at diagnosis has increased dramatically from a median of 16 years 2 decades ago to 34 years in the most recent period. However, surprisingly, despite this increase in age, we see a lower proportion with fibrostenotic disease, suggesting that these are not merely just catch-up cases. So could this indicate that historically only more severe and long-standing cases were diagnosed and now cases of shorter duration are being diagnosed? I mean, that does fit with the observation that in Denmark, we're not seeing the same leveling off as has been potentially observed in the pediatric data that I presented and in the data from the Netherlands. And then closer to home here in North Carolina from data Dellon collected, we again see this rapid increase in cases, an increase in mean age, but unlike what we observed in Denmark, we see a general increase in proportion of patients with fibrostenotic disease. So this increase in age distribution could indicate other potential factors are contributing to disease development or a possible catch-up in case diagnoses, with adult cases reflecting longer standing disease. In this next study, from a registry study of patients primarily from Spain, but also Italy, Denmark, and France, I think it says temporal changes in presentation of new patients over a decade of follow-up and reported no significant difference in age, but a significantly decreased diagnostic delay, an increased proportion of inflammatory as opposed to stricturing phenotype, and significant decreases in endoscopic severity scores. And further, the number of endoscopies prior to the diagnosis decreased. So generally suggesting this idea that newer cases were more likely to represent true incident cases. The challenge of estimating true incidence and prevalence is also hampered by access to care issues. And in this paper from Emily McGowan, they reported that risk of EoE in the pediatric population was lower in rural areas. But when we saw this paper, we were concerned that this might actually be explained by the fact that children residing in rural areas might have a much more difficult time getting diagnosed. So here we conducted a reanalysis of the data, this time accounting for distance to a pediatric gastroenterologist, and with this observed that access to care might have explained the differences in observed risk according to rural residential status. Okay, but it's not just about rurality. So in another study we just presented at DDW this year, and using data obtained through clinical sites supported through the Seeger Consortium, we sought to determine the frequency of biopsy conducted at an esophageal food impaction and differences according to patient factors to determine proportion of patients receiving timely follow-up with a gastroenterologist, which we defined as within 3 months of an esophageal food impaction, and again, according to patient factors. And then to determine proportion of patients receiving a repeat esophageal food impaction and according to patient factors. And here's what we found. So for biopsies, overall, we observed just 27% of patients presenting to the emergency department from these selected Seeger sites had documentation of having performed a biopsy at the time of the impaction. And as age increased, likelihood of getting a biopsy decreased. And similarly, as the area deprivation index increased, which is a measure of social deprivation in the neighborhood in which you live, likelihood of biopsy decreased. Having public insurance also decreased likelihood of biopsy. And for timely follow-up, 47% had timely follow-up, so within 3 months, and 61% had evidence of any follow-up with a gastroenterologist. Again, with higher area deprivation, decreasing likelihood of timely follow-up. And having a previous EoE diagnosis and getting a biopsy performed at the esophageal food impaction encounter actually increased likelihood of timely follow-up. And then for risk of repeat esophageal food impactions, 7.2% of patients had evidence of one or more repeat esophageal food impactions, but there were no statistically significant factors associated with this risk. So all of this just, again, highlights that we have yet to fully understand the true incidence and prevalence of EoE. So this last bit of epidemiology I want to touch on today is just prevalence within subgroups. And historically, EoE has been described as a disease occurring predominantly in white males. And this assertion has been perpetuated across sources. But what is the evidence for this, really? It turns out the evidence for predominance in whites may not be well supported by the current literature, at least not to date. And when we examine evidence on race, and in population-based studies, such as this one that use EHR data, we can see that while it appears that EoE is more common among whites relative to the source population, the degree of missing data for race makes this assertion somewhat unclear. So wanting to better understand the assumption of demographic differences perpetuated across the literature, we performed a systematic review and examined evidence of demographic differences using population-based studies that provided both the demographic distribution in cases as well as the demographic distribution in the source population that gave rise to these cases. And from the 50 studies we identified in our review, just 12 met criteria for inclusion. And of these, only one actually provided adequate data for examining racial distribution in both the case and source population. And this was a study conducted in New Zealand. So we therefore then concluded there wasn't compelling evidence to support that EoE is enriched in individuals of white race. We also examined sex differences and here found very clear evidence of a male predominance for EoE at roughly 2-to-1 ratio. And this was relatively consistent across all of the studies. So just to wrap this up, we see clear evidence of increased prevalence of diagnosed cases of EoE. And while the absolute number of newly diagnosed cases is still increasing, there is some limited early evidence of plateau in pediatric groups. Rurality may be intertwined with challenges in access to care, making it challenging to establish true incidence and prevalence. Encounters for food bolus impactions may be the initial healthcare encounter for some patients with EoE, so both provider and patient education is needed to support timely diagnosis. And then there's no evidence of differences in racial distribution for diagnosed cases, like clear evidence of sex differences for EoE. So just to summarize, the true incidence is yet to be determined, but we may be getting closer, and addressing physical and financial access to care challenges will support efforts to identify patients earlier in their disease trajectory and ultimately mitigate increased risk for complications related to longstanding disease. Thank you.
Thank you, Dr. Jensen, for that very helpful overview of the epidemiology of EoE. We have time for questions. I'll remind you to put your questions into the Q&A box. I believe you can upvote your favorites there. Just to get things started, I'll go ahead and ask a question. One of the challenges in understanding the epidemiology of EoE is the possibility of underdiagnosis. From your perspective, what approaches could help us better assess the potential underdiagnosis of EoE?
Yeah, it's a really, really tough question, but one possibility that would get maybe us a little bit farther down the road of understanding underdiagnosis is to look specifically at those sites where they have implemented interventions to increase the biopsy rate at the time of an esophageal food impaction. So there's been some terrific work in that space. Nimi Gonzalez has put together an interventional approach that I think Dellon also applied, and they went from roughly 30% to 80% of patients presenting with a EFI getting biopsied. And so with that, we can start to see what proportion of patients actually do have a true diagnosis of EoE in that particular case population. And we can start to extrapolate how many cases are we missing of more severe disease.
Thank you. That's very helpful. Let me shift to an epidemiologic pattern that you mentioned. The evidence for a plateau in EoE appears to be primarily in the pediatric patient population. Why do you think that is?
Yeah, I mean, I don't know that we can say for sure, but I suspect that what we may be starting to observe is that we're getting better at diagnosing patients readily, so that these truly are starting to represent true incident cases within that pediatric population. And then we still have quite a few adults that have been living with the disease for a very long time and have just adjusted to their symptoms. They have compensation that they have put in place for their symptoms and are probably still not getting diagnosed. But in the pediatric population, that there likely was some environmental exposure that increased the incidence of cases, but we're starting to see that plateau as we've reached capacity on exposure from that environmental exposure, and we're getting better at just diagnosing kids earlier.
Great. Thank you again, Dr. Jensen. If there's no further questions, we will move to our second presentation. No, sorry, give me a moment here. I see that Stephen Ackerman has asked, would access to minimally invasive screening tools be useful in addition to increasing performance of biopsy at time of EFI?
Yeah, I mean, right now, the only way to diagnose EoE is through an upper endoscopy and biopsies. But yes, absolutely. If we got to the point where we could identify disease process either through blood-based biomarkers or through less invasive approaches, it would be, I think, a game changer.
Great, thank you. Okay, any further questions? We'll go ahead and move to our next presentation. Thank you again, Dr. Jensen, very much. Our next speaker is Dr. Nurit Azouz, who will present Pathophysiology of EoE, Epithelial Protease Dysregulation, and Barrier Dysfunction. Dr. Azouz is an assistant professor in the Division of Allergy and Immunology and the Center for Inflammation and Tolerance at Cincinnati Children's Hospital Medical Center. Dr. Azouz leads a translational research program focused on the role of proteases and protease inhibitors in allergic and inflammatory diseases. Her work has helped uncover how dysregulated proteolytic activity in the esophageal epithelium contributes to tissue injury and inflammation in EoE. Her laboratory continues to investigate the molecular mechanisms underlying EoE pathophysiology with the goal of identifying new therapeutic approaches for EoE and related allergic diseases. It's my pleasure to welcome Dr. Azouz. Dr. Azouz, I'll turn it over to you.
Thank you so much for the kind introduction, and I'm very grateful to be here today and for the opportunity to talk with you. All right. Today I will talk about the pathophysiology of EoE and the role of proteases and protease inhibitor. As you all know, I don't know why my slides are not advancing. Okay. We know that EoE pathogenesis is a complex disease, involves multiple cell types, and it involves the infiltration of immune cells to the esophagus, such as eosinophils and mast cells that induce tissue damage. We also have Th2 cells, including food antigen-specific Th2 cells that undergo clonal expansion and persist within the tissue. And we know today that the main source of IL-13 in the esophagus comes from mast cells and Th2 cells. So the importance of IL-13 in EoE is highlighted by the efficacy of biologics that target the IL-13 pathway. IL-13 is a key player in EoE pathogenesis, affect multiple cell types, including epithelial cells. So the esophageal epithelium is a stratified barrier that normally prevents from food antigens to penetrate the underlying tissues. IL-13 and then other external and internal stimuli induce major transcriptional changes in epithelial cells, including differentiation defects that disrupts the formation of differentiated layers of epithelial cells that form tight junctions between them and therefore are critical for the barrier function. So when this barrier is impaired, epithelial cells become activated. They release pro-inflammatory mediators such as CCL26 that attract eosinophils to the tissue, and also the alarmins IL-33 and TSLP, which are major driver of type 2 immunity. These alarmins act on multiple cell types, on dendritic cells, and then directly or indirectly on mast cells, basophils, Th2 cells, eosinophils, and ILC2s. Consequently, these immune cells activate fibroblasts. Those fibroblasts release, produce more collagen and extracellular matrix proteins, leading to esophageal stiffening and strictures. And in addition, eosinophils and mast cells reside in multiple tissues, including the smooth muscle compartment. They release TGF-beta, and they're also granule content are affecting smooth muscle dysmotility in the tissue. So overall, we have this positive feedback loop where we have immune cells that activate epithelial cells that further exacerbate inflammation and cause this vicious cycle in EoE pathogenesis. And I'd like to highlight your attention here to the upstream regulation in the epithelial integrity that conserved between a fine balance between proteases and protease inhibitors. So over the last few years, we identified SPINK7, which is a serine protease inhibitor that is highly expressed in the esophageal epithelium to be downregulated in the esophagus of EoE patients. And we found that with the loss of this protease inhibitor, we have proteases that are unleashed in the esophagus. This includes the downstream target of SPINK7, the serine protease kallikrein-5 that we identified. Kallikrein-5, when it's unleashed, it will start cleaving junctional complexes between epithelial cells, including desmoglin-1, causing barrier impairment. So you can imagine that if food is rapidly flow through the esophagus, when we have this barrier breach, we can have food antigens penetrating the underlying tissue, encountering immune cells and inducing type 2 immunity. We also found that kallikrein-5, together with other serine proteases, will activate the protein as activated receptor 2, PAR2. When PAR2 is activated on epithelial cells, these epithelial cells will release massive amounts of cytokines and chemokines, including TSLP. So after identifying this mechanism, we ask if we can reverse the uncontrolled proteolytic activity by delivering protease inhibitors to the esophagus and then inhibit some of the downstream effects such as barrier impairment and innate immunity. So the first protease inhibitor that we tested was the alpha-1 antitrypsin, A1AT. First, because we have shown that A1AT inhibiting kallikrein 5, which is unleashed in the esophagus of EoE patients. It was also readily available for us because it's an FDA-approved drug for the treatment of alpha-1 antitrypsin deficiency. So the experiment that I want to show you over here, we took esophageal epithelial cell line and we silenced Sphinx7, or we used a non-silenced control. We grow the cells and differentiate them in a system of air-liquid interface. And then we measure the trypsin-like proteolytic activity of the supernatant of the cells. And we are hypothesizing that with the loss of Sphinx7, we will see increased proteolytic activity. And indeed, that's what we have observed. So you can see here the gray bars. These are the Sphinx7 silent cells. Without this inhibitor, we have increased proteolytic activity. When we deliver to those cells A1AT, A1AT was able to dose-dependently inhibit the proteolytic activity in the supernatants. We also analyzed the histology of the cells. If we look at the control cells over here, they form a very intact barrier. You can see here the cell nuclei, and then on top you will see the differentiated layers that are here in pink. These are the squamous cells that are very important for the barrier function. When we silence Sphinx7, we see differentiation defect. We don't see these differentiated layers on top, and we also observed intercellular dilated spaces. However, when we delivered A1AT to the Sphinx7 silent cells, the spaces between the cells was decreased, and also we saw normal differentiation with the formation of the differentiated layers. So we suggest that A1AT administration improves the epithelial integrity. We then tested the effect of A1AT in vivo. We used an OVA-induced EoE-like murine model. And during the challenges with OVA, we treated the mice either with human serum albumin as a control or human serum A1AT. And then at the end of the challenges, you can see the section through the esophagus with the MBP staining that correspond to eosinophils. You can see eosinophils accumulate in the esophagus after OVA challenges in the albumin group. But in the A1AT group, we see decrease in eosinophilia. And over here, you can see the quantification where each data point represents the number of eosinophils per one section per mouse. So you can see the significant decrease in the number of eosinophils in the A1AT treated group compared to the albumin group. And we suggest that A1AT attenuates eosinophilia in an allergic murine model. So how does A1AT works? And A1AT belongs to the serpins superfamily. This is a superfamily of serine protease inhibitor, and they have a very interesting mechanism of function. So the serpin have a reactive serpin loop that is recognized by proteases as substrate. So the protease will cleave the serpin in the reactive loop, but this is actually a bait because now the serpin will covalently bind the protease. So we're getting a complex of serpin with the protease, and the serpin masks the active site of the protease. So there is no access for the substrate. So this protease is non-active, and the formation of this complex is irreversible. So it has been known for many years that serpins have proteolytic inhibition capacity, but they have also additional role. So once they form the complex with the proteases, they also expose their C-terminal. The C-terminal is highly conserved between all the serpin superfamily, even though it's not part of the inhibitory domain. But those sequences contain the binding site to scavenger receptors. Most known, the LRP family, the LDL receptor protein that then will bind the serpin in the complex with the protease. They will induce endocytosis and elimination by lysosomal degradation. And it has been shown in several disease models that activation of LRPs by the serpin and protease complex can induce different signaling pathways in the cells. This can be either pro-inflammatory or anti-inflammatory. So Eman Sajib, a PhD candidate in my lab with the help of Kaylen, a research assistant in my lab, asked, what is the mechanism of serpin's function in the tissue, specifically A1AT? Is their function mediated through proteolytic inhibition or agonistic function through activation of LRP, or maybe both? So to tackle this question, we first tested how much the inhibition of proteolytic activity is necessary. So I have shown you before that in EoE we have proteases that are unleashed. They will cleave and activate part 2 that will lead to TSLP production. So if we deliver protease inhibitor like A1AT, we can inhibit the downstream events that are unleashed by the proteases. If the inhibition is necessary, then any protease inhibitor will work pretty similar to A1AT. So we wanted to investigate the role of small molecule inhibitors. So here you can see the light gray here. It's a 3D structure of a protease, and small molecule here in the colorful molecule that you see will bind to the active site of the protease. So it will inhibit the proteases like kallikrein 5, but it doesn't have a C-terminal that will activate LRPs. So we tested the effect of small molecule inhibitors. So in this experiment, we took esophageal epithelial cells with SPINK7 knockouts or control. We stimulated the cells with poly-IC, and you can see that we have production of TSLP that is increased in the SPINK7 knockouts and not in the control cells. And then if we deliver A1AT, we can inhibit this TSLP production. We then use 2 small molecule inhibitors that are very potent in inhibiting kallikrein 5. And those 2 small molecule inhibitors did not inhibit TSLP at all. So those small molecule protease inhibitors don't have the same effect as a full-length A1AT. So that made us suspect that maybe the C-terminal of the serpin is necessary for inhibiting the innate immunity. So at the next experiment, we took small molecules inhibitors, and this time, instead of comparing them to the full-length serpin, we took the C-terminal of the serpin. So sp16 is 16 amino acids derived from the C-terminal of A1AT, and they contain the binding site for LRP1. 7G is a new analog of sp16 with greater stability. So first, we tested what will be the effect of those small molecules and peptides on the proteolytic activity. And you can see that the small molecule inhibitors are very efficient in inhibiting proteolytic activity. And as expected, the peptides derived from the C-terminal of serpins, they do not inhibit proteolytic activity at all. And then when we measure TSLP production, the 2 small molecules did not inhibit TSLP, and then our peptides, sp16 and 7G, were inhibiting TSLP, completely blocking TSLP production. So we know that serpins can inhibit proteases and downstream signaling, including TSLP production, but also serpins are needed to interact with the proteases, activate LRPs, and then inhibit TSLP production. And not only TSLP production. So in this experiment, we stimulated the cells with IL-13 to produce CCL26 production, and then 2 concentration of sp16 were inhibiting the CCL26 production. And we ask if this is dependent on LRPs. So at the following experiment, we triggered the cells with poly-IC to induce TSLP production, and then sp16 derived from A1AT completely blocked TSLP production. But to ask if this is mediated through LRP1, we used an antibody against LRP1. So here you can see a scheme of LRP1 structure. LRP1 has 4 extracellular clusters that can bind serpins. 2 of those clusters are the most famous clusters that bind different serpins in complex with their proteases. And our antibody bind one of these clusters. So the other clusters are still open, but still when we add antibody against LRP1, we are attenuating the effect of sp16, and the cells produce low amounts of TSLP. We also used another inhibitory molecule, RAP, which is a protein that will bind all 4 clusters of LRP1, but also extracellular domains of other LRP members. And we see even greater attenuation in the SP16 effect, and we see greater TSLP production. When we combine the anti-LRP1 and the inhibitory molecule, you can see that we have even greater production of TSLP. So we suggest that SP16 works via LRP1, at least in part. We were also very interested about the downstream signaling that serpins mediates. So we perform a phosphoarray to screen which kind of molecules are activated by serpins, and we identified c-Jun. So here you can see that c-Jun is getting phosphorylated in 2 phosphorylation sites when we activating the cells with SP16. And we also showed that this is mediated through LRP1. Here you can see phosphorylation of c-Jun by SP16, and when we're using either the inhibitory molecule or the antibody, we are starting to lose the phosphorylation of c-Jun, and you can see here the quantification. So we conclude that SP16 induces c-Jun phosphorylation in an LRP-dependent manner. So I have shown you today that we have this positive feedback loop between epithelial cells and immune cells, and serpins and proteases, they have a very complex mechanism of regulating the epithelial integrity and innate immune reactions. So when we have epithelial cells that are triggered by different stimuli like IL-13, or maybe they have uncontrolled proteases that cleave PAR2, we can deliver serpins that can inhibit the uncontrolled proteases, but those serpins are also interacting with the proteases, activating LRPs. LRP downstream activates c-Jun phosphorylation, and then we are heavily focusing on what is the mechanism of action of c-Jun activation and phosphorylation, and if it has a role in inhibiting the pro-inflammatory cytokine production by epithelial cells. So thank you all for listening. I'm very happy to be here today. I want to thank also my mentors and collaborations, and especially the lab members and our fundings.
Thank you very much, Nurit. We've got some questions. That's very interesting. Let's see, the first one is from Alfred himself. From a translational standpoint, what do you think would be the biggest challenge in moving from these mechanistic findings towards a therapy that restores the protease inhibitor balance in esophageal epithelium?
Yeah, so that's a great question. I think like when thinking about this like positive feedback loop, inhibiting any arm there would be very beneficial. And when looking specifically on protease and protease inhibitors, so of course proteases, they also have their physiologic function, and they are imported in many processes like differentiation of the cells, the squamation, shearing of old cells from the tissue. So it takes a very fine balance. So we don't want to inhibit too much the proteases. We want to retain their physiological function, but only inhibit the uncontrolled proteolytic activity. So I think this will be pretty challenging thing to do. I hope that maybe with the small peptides that will only inhibit the downstream signaling but will still retain normal protease activity, we can test that and see if that will be effective.
Okay. Just keep on throwing in all the questions, everyone out there. In the meantime, I'll ask a question. Nurit, that was, fascinating stuff, very elegant. Since STAT6 and poly-IC signal, the signaling pathway for IL-13 and poly-IC is so different, yet you find a commonality of signaling inhibition, and it may converge on c-Jun or AP1 signaling. How do you, put this together? And how specific is this? If you're showing 2 unique signaling pathways are blocked by this pathway, are you seeing that this is a broad effect, or you actually, what is the specificity for this as you uncover the speculation around the mechanism?
Yeah, it's an important question. So we still don't know. We need to further investigate that. So it can be that c-Jun is very general and very downstream, and it can inhibit many pro-inflammatory signaling. But we still need to investigate that and use different stimuli to see how specific it is.
Okay, let's see. We have another question, which is, can you talk about your recent OVO-L1 work and how does the OVO-L1 sphinx axis fit into the broader model of epithelial barrier dysfunction?
Yes, so we recently published that OVO-L1 is a transcription factor that is upstream regulating sphinx 7 expression. Because one question that was very interesting for us is why does we have loss of sphinx 7 in EoE, and if we can control the expression of sphinx 7, so we will inhibit the proteases by endogenously overexpress sphinx 7. So we performed screen of transcriptional factors, and the only one that we identified was OVO-L1 that regulates sphinx 7. And we also found that OVO-L1 has upstream regulation. So IL-13, for example, can induce the exit of OVO-L1 from the nucleus to the cytoplasm, so then it will not be active and will not induce sphinx 7 expression. So it's a very complex regulatory network.
Thank you. Fascinating work. We're going to move on to the next speaker. Alfred, I'll let you introduce Evan.
Thank you. Our final speaker is Dr. Evan Dellon, who will present New Frontiers in EoE. What does the future hold? Dr. Dellon is a professor of medicine and adjunct professor of epidemiology at the University of North Carolina School of Medicine, where he also serves as director of the UNC Center for Esophageal Diseases and Swallowing. Dr. Dellon completed his fellowship training in gastroenterology at UNC, where he also earned an MPH in epidemiology. His research encompasses the epidemiology, pathogenesis, diagnosis, treatment, and outcomes of EoE and other eosinophilic gastrointestinal diseases. Dr. Dellon's work has been instrumental in advancing the understanding and clinical management of EoE with the ultimate goal of improving patient care and outcomes. It's my pleasure to welcome Dr. Dellon. Dr. Dellon, I'll turn it over to you.
Thanks so much, Alfred, for that introduction. Thank you for the invitation to be here. This is a tremendous conference, and I'm very happy to be here and talk about sort of new frontiers in EoE. I have a number of disclosures. They're not particularly pertinent to this talk, but I do mention some therapies with companies that I consult with. The main disclosure for the talk, though, is it's tough to make predictions about especially about the future, like Yogi Berra once said. And so you can't hold me to any of these, but maybe some of them will be reasonably accurate. So what I wanted to do in the talk today is to first talk about where we've been, and that helps us, I think, contextualize where we're going. And where we're going, I'll talk a little bit about diagnosis and monitoring, just very briefly on pathogenesis and epidemiology, since you've heard two great talks about this, and then talk a bit about treatment. And I think you'll see that for some things, the future actually may be now. So first of all, there's been an enormous rise in EoE publications and knowledge. And I think the prior two talks gives you just a little bit of a sense of this, but I just wanted to mention that I think a lot of this is in part because of the really collaborative nature of the fields, spanning basic translational clinical research across multiple disciplines and across pediatrics and adults, and research involving patient advocacy groups and patients themselves. Platforms like this from professional societies like IES and others has really been able to foster this this type of enormous increase in knowledge. So with that, how do we think about diagnosis of EoE? So if we look back over a timeline over the last several decades, EoE is still a young condition, with the first case series of what we would recognize as the modern aspect of the disease really only being reported about 30 years ago. And it wasn't until 2007 that we had our first guidelines. And then there's been a rapid increase in multiple guidelines, most recently with American College of Gastroenterology updated guidelines, in 2025, and we're awaiting this year publication of European and EUREOS guidelines, which are quite concordant with the most recent US guidelines as well. So there's been a tremendous amount of knowledge. And so where do we stand with our diagnostic criteria? Well, we have a pretty standard algorithm now where we have a clinical presentation in a patient that's suggestive of EoE. This leads to upper endoscopy with biopsy. If you find at least 50 EOs on the biopsy, then you do a step where you have to evaluate for non-EoE disorders that could cause esophageal eosinophilia or the clinical picture. And then ultimately you can diagnose EoE. And even though this has been pretty standardized and there's been no real major changes to this over the past 8 years or so, there are things that potentially are missing. So when we think about the clinical presentation suggested of EoE, all EoE is not the same EoE. There are phenotypes and endotypes. The second step is the endoscopy. As you heard already, this is kind of burdensome. And will you always need an endoscopy to diagnose EoE? And what does this third step actually really mean? Like, do we have to do this? Can it be done more easily? So let's go through a little of these missing things and think about the future in some ways for this. So phenotypes and endotypes, I think as this audience probably knows, a phenotype is an observable feature in an individual. And in EoE, there are multiple types of phenotypes. You could think about allergic versus non-allergic patients, pediatric versus adult onset, fibrostenotic versus inflammatory, mucosal versus transmural, patients with and without family history, and then things that are, treatment refractory patients or patients who do and don't have connective tissue disorders. And so all of these may have impacts in diagnosis and outcomes. And then there are endotypes, which is a subtype of a condition defined by a biologic mechanism as opposed to those observable features. And the Seeger group, led by Tetsuo Shoto, was able to demonstrate for the first time that there seem to be 3 molecular endotypes of EoE that correlate with some of these clinical phenotypes actually in a nice way. So for example, EoE E3 seems to be more fibrostenotic, whereas type EoE2 might be more steroid refractory. But there's other ones also. You might have a T2 high versus low. You might have patients with abnormal barrier function versus not, as we just heard, or there might be genetic markers. So why do we really care about this? Because I think understanding these phenotypes and endotypes can help with diagnosis, mechanisms of disease, ultimately treatment selection, and prognosis. And I'll talk more about precision medicine in EoE in a little bit. Now, when we do that next step, when we go on to the endoscopy, how do we optimize this? Well, for many years now, we have the EoE endoscopic reference score, or EREFS, derived by Iko Hirano and his colleagues. And these are pictures on the left here of a patient with newly diagnosed EoE before treatment. And you can see clear endoscopic findings, including exudates, edema with loss of vascular features, the rings, the furrows here going up and down the esophagus. And this is actually narrow, and there's no clear stricture in this picture. And then after treatment, you see resolution. The esophagus looks normal. And so, where are we going with these endoscopic features? Well, now there's a question of, can endoscopic features actually be a primary outcome, a biologic outcome in EoE? You're looking at the disease improvement, right? And you can see between these two pictures that you might not need a fancy scale or biopsies to know that that patient on the right after treatment is better because the esophagus is normal. Similar in eczema, right? The skin is normal, that medicine is working, that patient is better. And then in addition, we have EndoFlip. This is a catheter-based technology where we can measure the motility and distensibility or mechanical properties of the esophagus. And this is an example of EndoFlip in an EoE patient. And what we're looking at here is a balloon catheter in the stomach coming proximally through the esophagus. This is the esophagogastric junction where there is likely a stricture. The higher, the more intense the red, the more narrow the area. And coming up into the body of the esophagus, this is a narrowed esophagus. And as you look forward in time, this is time moving this way, you're seeing no motility here, and you're seeing just a very narrow esophagus that doesn't distend. And so where are we going with this? Well, people have now used this tools, in particular, Khalees Menard-Ketchard and Amanda Muir have actually started to measure in children what is the normal size of the esophagus? How does the esophagus change with growth? And how does that impact ostensibility? And the Northwestern group has proposed a physiomechanical model of esophageal function in EoE, where you have multiple possible patterns of motility overlaid on the distensibility. And some of these might have prognostic implications, and particularly these patients with that non-reactive fibrostenotic, like that first image I showed you, may have decreased response, for example, to proton pump inhibitors. And so we're developing more advanced ways to assess the esophagus during endoscopy. Now, what about the opposite of this? And this goes to one of the questions that was asked as well. What about less invasive ways to assess the esophagus? Well, transnasal unsedated endoscopy is here. It is at multiple centers now, and it's becoming more commonly used in children as a way to minimize sedation, make procedures more efficient, and less expensive. But this is still an endoscopy. It still requires a tube going through the nose. There's discomfort associated with this. And so there are less invasive ways that sample the esophagus, one of which is the cytosponge, which is a capsule that you swallow that once that capsule dissolves, it encloses a spherical cytology brush. This is swallowed. The capsule dissolves after 7 minutes. This brush then is pulled out of the esophagus. It collects essentially a biopsy sample that can be analyzed in regular pathology labs. This has limited availability right now in the US. It's actually used now routinely in the UK for screening for Barrett's esophagus. And so there is optimism here, I think, in the US that this will be looked at again and maybe in a role for monitoring. What is available right now and is having increasing availability is the esophageal string test. This is a capsule that contains a string, and the capsule deploys down the esophagus and releases the string. The capsule dissolves. The string sits in the esophagus for an hour, absorbing inflammatory factors related to EoE. And this also now has a role for monitoring and is available in a number of centers. Now, these are not right now for diagnosis, but they are having increasing utility and are being studied more. What about truly non-invasive tests for EoE? For example, blood tests. Well, if you do a PubMed search, there's more than 120 studies on blood-based biomarkers. There's also saliva biomarkers, breath biomarkers, even stool biomarkers. And this is just an example of one that was done by Jared Sninsky when he was a fellow at UNC, where in the blood gene expression, the IL-5 receptor alpha gene, well known to this audience, was actually the most highly upregulated gene in the blood and was quite a good diagnostic biomarker for EoE. And there are multiple other types of assessment agents, including mucosal impedance, which measures barrier function in terms of resistance and leakiness of the barrier. There are tethered capsules that can give visualization of the esophagus. And there are also unique spectroscopic signals in the esophagus. So all of these might be minimally or less invasive tests for EoE. But there's a lot of these. Many have shown initial proof of concept. Why haven't these come into clinical practice? What's needed to implement these biomarkers in the future? And I think this is where the hang up often comes. And this is something that we need to address as we go forward in the future of EoE. So first of all, we need prospective validation studies. Many of those papers looking at new biomarkers are case control studies looking in biobanks. And these are great, but these are previously collected samples. It's generally a small group of patients, and it's a way to do discovery work. But what we really need are studies where you measure the biomarker pre-treatment, you measure it post-treatment in the same individuals, and validate that, yes, this actually is responsive to treatment and matches our other treatment response outcomes that we use in EoE. And then we also need confirmation of a biomarker, ideally in a directed care type of trial. So you might randomize patients to biomarker-guided treatment and monitoring versus standard endoscopy-guided treatment and monitoring. And this would be the gold standard that shows that a biomarker works just as well as our current standard of care. And many clinicians would require this level of evidence to adopt something. And, obviously what's really needed is funding, but it's challenging because these types of studies that I've just described, these are important, but they're also large and expensive, and they are not novel in the sense of discovering new genes, new markers, etc. And so there's often an issue with innovation here, but these are so important for confirmation and implementation that I think we can make a strong case for funding these kind of studies in the future with our most promising biomarkers. Now, moving to that third step in the diagnostic algorithm, how do we make a positive diagnosis? Well, there's plenty of work on this, including landmark work by Dr. Rothenberg and his colleagues that show that you can molecularly diagnose EoE with almost perfect accuracy, looking at the EoE transcriptome. There's other tests that you can use also looking at tissue-based markers and immunohistochemistry. And so we have ways to make a positive diagnosis of EoE without necessarily that third step of having to think about what else may be going on. We could actually test and make sure none of those things are going on. Now, when we think about pathogenesis and epidemiology, I didn't want to recapitulate the points that have already been made, but I would note that this rapid rise in EoE, as Dr. Jensen mentioned, points to changing environmental factors. And the key thing here going forward is understanding the interaction between these environmental risk factors and triggers and the underlying genetics in the context of esophageal barrier function. Because the key issues are that we cannot answer what causes EoE in an individual person. And we can't answer, is there a way to prevent that causative nature? And without this, we can't move forward to understand really how to cure EoE. So these are key points where hopefully pathogenesis and epidemiology work will merge to help us with our clinical algorithms as well. Now, for the last part of this talk, let's go through where things stand now and moving forward for treatment. And when we look at the same timeline for EoE treatment, I think you get a sense there's sort of a crescendo going over to the right side of this slide here, where the first reported treatment for EoE was elemental formula, also demonstrating that EoE is a food allergy-driven condition in the vast majority of patients, onto topical swallowed corticosteroids, and then moving into biologic trials. And as you can see here now, we're at the point where we have the budesonide dissolvable tablet approved for treatment in Europe. We have dupilumab approved for treatment across the lifespan. We have finally in the US the budesonide oral suspension, and we're expecting readouts for other clinical trials, including an anti-TSLP agent later this year. And if that's positive, hopefully that will move forward to our second biologic that's been approved. So this increasing amounts of treatment has been good news for our patients for sure. And this is really driven by our knowledge of EoE pathogenesis. This is a much more simplified schema than what we were just looking at in the prior talk. But I put this up not to necessarily go through the EoE pathogenesis in detail here, but to show you on the next slide here that because of our knowledge of EoE pathogenesis, we have novel treatment targets at essentially every step in the pathogenesis. And, we just heard about the alpha-1 trypsin inhibitor, and pretty much at every step of the cascade here, there's something going on. And this schematic is things that are in blue are biologics, things that are in black are small molecules, and if it's got the yellowish orange box, that's already gone to phase 3. And so this is really a highly exciting thing. And these agents may not work just in EoE, they may work in non-EoE ages as well. So what's in our immediately pipeline coming down here in the future? Well, we have 2 anti-TSLP agents, one in phase 3, tezepelumab, and one in phase 2, silrucatug. And we'll hear about both of these hopefully in the coming months. Has been anti-KIT antibody barzolvolimab that just had a press release with results, although they haven't been presented yet, that showed that eliminating mast cells actually did not seem to help EoE. And so an important experiment where we're learning more about what triggers to hit. And then you can see here there's proof of concept for anti-IL-15, anti-integrins, phase 2 data on modulating the sphingosine-1 phosphate receptor and using an immunoregulatory protein. These both have proof of concept. JAK inhibitors have proof of concept. There's a PCAB, we heard about this one already, a PCAB study that is going on now that has recently completed enrollment. And most recently, a long-acting anti-IL-13 has been reported that a program will be starting. So the future is very bright and quite full with things in the pipeline and many more targets actively being identified. And so with that, yeah, there are more than 45 active clinical trials for EoE. And so with that, we have our current treatment algorithm for EoE, which recommends that patients are always assessed for fibrostenosis and do they need esophageal dilation and started on an anti-inflammatory therapy. The current dictum is to use shared decision making to decide if it's pharmacologic therapy or diet elimination therapy. You select a therapy and assess response. At the same time, you're reassessing if there's strictures that need to have dilation. If there's a response, you maintain those treatments long term. If there's not a response, you may change original therapies or step up to dupilumab, and then again assess response. So this is kind of the overall trend, and you can see here that it's sort of a one approach for every single patient with a heavy reliance on shared decision making. And I think this algorithm works well now for our current patients and for the current kind of treatments we have. But going forward, I think many of us are thinking about how do we personalize our treatment above and beyond a discussion of preferences and shared decision making. And this is happening now. What you need for this is predictors, right, of treatment response, and we have these. So on the PPI side, a number of papers show that the CYP2C119 genotype, whether you are a rapid or ultra rapid metabolizer, can impact your PPI response, as can STAT6 variants. And then on the steroid side, there are a number of clinical factors that predict lack of steroid response. And most recently, Dr. Jensen has also published with colleagues a panel of epigenetic markers that has been validated in an independent population that predicts response to topical corticosteroid therapies. So we have the tools that we can potentially use. We may even have these tools for biologics. This is work from Julie Dunn, Dr. Rothenberg, and the Seeger group that shows this heterogeneity of type 2 cytokine expression in the esophagus. And as you can see, these are all active EoE patients. Their eosinophil levels are high here. But there are things that are different. There are some people who are very high in IL-5. There are some people who are very high in TSLP, some who are higher in IL-13, some who are higher in mast cells, and these don't all necessarily overlap. And so this may also suggest that some of this testing could impact what biologic you might choose for a given patient. And then even for dietary elimination now, there is quite a lot of work going on to figure out if there's a way that we can figure out individual food triggers in individual patients, whether it's looking at injecting allergen into the esophagus and looking at a specific response here, like in this first paper, whether it's looking at food-specific antibodies or T-cell stimulation assays. These are all things that are being tested and may help us optimize diet elimination for patients. In terms of, so we have these, and it's similar to biomarkers, right? Why aren't we using these? Well, we need data from clinical trials to show that these work. And I think what we're going to see is a transition from placebo-controlled trials, like you see on the left with treatment A and placebo, to comparative effectiveness trials like treatment A and treatment B. And actually at DDW last month, there was the first, I would say, modern randomized trial comparing PPIs and an esophageal specific topical steroid. So it's pantoprazole versus a budesonide dissolvable tablet at the doses we currently recommend to use for EoE, and showing that the topical steroid was about twice as effective as the PPI. And so those are the kind of data that really can impact our treatment algorithm and our guideline recommendations. There may also be a transition to pragmatic clinical trials, and these are examples from New England Journal of non-GI pragmatic clinical trials. But here the idea is to use outcomes and assessments that are available in practice as a way to do large-scale, very efficient trials without necessarily needing all of the typical clinical trial infrastructure that's so cumbersome and expensive that we think about in these traditional placebo-controlled trials. And then finally, an increasing focus on patient-centered outcomes and also patient-powered research networks. And we have one for these in EoE and EGIDS. And I would refer you to egidpartners.org. If you're listening, go ahead and log on. This is a patient-powered research network. It's an online cohort that Elizabeth Jensen, myself, several other researchers in collaboration with our patient advocacy groups have founded. It's got almost 1,000 people, patients registered worldwide. They have EoE, other non-EoE EGIDs, and it also recruits controls, patients without EoE or EGIDs. And we focus on ideas for research from the patients. And patients can suggest research topics. We feed them back the results. On this website, you can see real-time data in aggregate, not individual, of course, about the symptom profile, the most common symptoms, the most common treatments, the locations around the world, the breakdown of cases. And so this is something that I think can be very useful. And this may also be able to be a way that we can use as an adjunct in clinical trials. So if you haven't seen this, check it out, ask your patients to check it out. And this hopefully will continue to grow as we go forward in the future. So in summary, for the near future, this is really here. We've got approved medications in the US and around the world. We've got esophageal specific topical steroids. We've got one targeted biologic approved and other targeted biologics and small molecules being actively studied. There are less invasive monitoring techniques coming online. Transnasal endoscopy and the string test are actually in many centers now. And hopefully these will all lead to decreased diagnostic delay with wider recognition of EoE, both among providers, but critically, I think, among patients. We can't diagnose a patient who doesn't recognize their symptoms as something to come and get checked out and get diagnosed. And as they come in and we diagnose them earlier, we can get quicker access to treatment and prevent complications. And then in the longer view, there should be individualization of therapies via phenotypes, endotypes, and predictive testing. I think we'll see this move towards comparative efficacy studies so we can position different therapies in different places for different patients. Hopefully our overlap of the epidemiology and pathophysiology will help us understand the cause of EoE, leading to screening and prevention, and of course, ultimately cure. So where might we be in EoE in 10 years from now in 2036? Well, you still got to have the clinical suspicion. The patient still got to have a symptom and present to the doctor or healthcare provider. Are we going to get away from upper endoscopy with biopsy for definitive diagnosis? Probably not. There may be a screening step in between to get people to an endoscopy sooner. But because so many other GI causes can be responsible for symptoms of dysphagia, abdominal pain, heartburn, you still often may have to take a look, at least right now. And of course, if there is a stricture, you're going to need dilation. You need to do an endoscopy to do dilation. On the other hand, once you confirm that EoE diagnosis, I think this is going to be confirmed in a positive way. Probably still with histology, but perhaps with tissue markers or a genetic profile or some other novel test yet to be developed. So you've got your confirmation of the diagnosis. Then I think what we're going to end up doing is non-invasive baseline testing. That lets us be sure that our non-invasive test correlates with our endoscopy. And then we do a phenotype endotype assessment. And then we do a predictive treatment assessment. And all of these can be done potentially upfront in the same diagnostic kind of session. And with that predictive treatment assessment, of course, taking into consideration patient preferences, we'll be able to decide, is it a PPI? Is it a topical steroid? Is it dietary elimination? Or is it a biologic? And probably this is going to also include some severity assessment for the patient. But if you have a patient who's severe and who's an ultra rapid PPI metabolizer and who has a narrow caliber stricture, those are patients who may not respond to PPI topical steroids. And maybe that's a patient who's going to be biologic targeted. And then within that group, you might pick which biologic based on other biomarkers. Then you're going to probably do non-invasive response monitoring and then individualize your therapy going from there. So it may take me out of the loop here as a gastroenterologist in terms of performing a lot of endoscopies, but this may be where we're going. And we have a lot of these pieces in place already. So thank you so much for being here and letting me give this talk. If you're in Chapel Hill, come say hi to me, and happy to take questions. Thanks so much.
Thank you, Dr. Dellon, for that thought-provoking look at the future of EoE care. And we do have time for questions. I see that we have an audience question from Andrew for Dr. Dellon. Is detergent exposure contributing to the increase in EoE, and what is the pathogenesis?
It's a really good question, and I think there is an idea that there are something in the environment is changing, right? And so Alfred, you could probably answer this question just as well as I can because it's your and Ben Wright's really interesting novel work showing that if you just put a detergent into the drinking water of a mouse, they develop EoE. And I think it's, a very plausible hypothesis, but I don't think it's necessarily the only thing. Right, the SDS detergent that you guys studied, it's in toothpaste, for example. Well, nearly everybody in the country brushes their teeth with a toothpaste that contains that detergent. But only a small fraction of those people get EoE. So why is that? Well, it's probably because the detergent, which we think would disrupt barrier function, maybe allow easier antigen ingress, starting that inflammatory cascade, there probably needs to be some other genetic predisposition or second or third hit to make that happen. And that's, I think, the case with a lot of the risk factors that are coming up. So early antibiotic exposure, for example, the same thing. A zillion kids get early antibiotics, but only a small subset of those may go on to get EoE because of that antibiotic exposure. So what's that other hit? And it may be an acquired barrier defect, but that I think still remains to be understudied. But I think what's interesting with the barrier stuff, this is completely speculative, of course, but if you go back to when the first EoE cases might be, it probably is in like the 1950s, 1960s. And one of the things that was developed around that time was dishwashers and these soaps that have, barrier disruptors and detergents in them. And the detergents have been linked to IBD, they've been linked to other atopic conditions. And so there may be something to that. And, certainly we didn't have dishwasher pods and all these other adherent detergents, but I think it's only probably part of the story. So maybe a longer answer than you wanted, but very interesting.
Thank you. That was great. And we have another audience question. Jerry asks, how close are we to an FDA-approved treatment option for non-EoE EGIDs?
That's a good question. I think, unfortunately, we're not that close. The Seeger group has just finished, and hopefully it will be out soon, the paper, a study of dupilumab in eosinophilic gastritis. And it was essentially a phase 2 study that was initiated by the consortium, and it had very positive, very encouraging positive results. The issue is that it was a very small study, about 40 patients, and there are a lot of hurdles to get over. I think we have a lot of potential medications and potential targets. I think the rarity of those conditions makes patient recruitment difficult. It makes understanding what the exact regulatory outcomes that we need to try to achieve difficult, including validated patient reported outcomes that are disease specific. And so I think we're ahead of where we were with EoE, sort of at the same research maturity in the field, let's say. But I think it's still a little bit of a ways off. I will say that it's something that people in the field are very interested in, know about, realize that it's a major gap. The FDA is also engaged, and so hopefully we can move things forward a bit faster than our first drugs that were approved for EoE.
Thank you. I want to follow up on that with a question of my own about therapeutic approaches for EoE. You highlighted several that are in various stages of development, including some that are already in later stage trials. Which do you think are most likely to meaningfully change clinical practice in the coming years?
I think there's a few trends. I think one trend is that we know a lot more about what targets to select now. We've done experiments now in randomized clinical trials where we've eliminated every eosinophil from the body and have shown that, surprisingly, I think to everybody in the field, that really didn't work for EoE. You had patients not feeling better. You had the endoscopy not looking better, even though there were no eosinophils there. And similarly, like I just mentioned, for mast cells. And so I think targeting the effector cells is probably not the way to go for in EoE because there's so much upstream pathogenesis and tissue damage still going on. So I think that's one trend in selecting sort of upstream targets. The second thing is probably trends for injectable medications of looking for medicines that are longer acting to improve the clinical experience. So there's not such frequent injections. I think that's probably another trend. And then the last thing is there are several, well, maybe more than one last thing. I think the next thing is looking for small molecules, right? We don't have any non-PPI, non-steroid targeted small molecules for EoE. And there are some that are quite promising. We have now proof of concept of modulating the S1P receptor. That's a medicine, even though the effect in that particular trial was relatively modest, it was still positive for a phase 2. And that medicine sequesters lymphocytes, including eosinophils, but also, say, T cells in the lymph nodes. It doesn't let them get to the target organ to create the local inflammation. So, a strategy along those lines may be effective, perhaps for maintenance therapy, maybe not initial therapy in a very inflamed patient. There's other medications that seem to be able to potentially induce immune tolerance, and so that would be a clinical game changer if those things come to light, and that's what that chaperone protein derived from the tuberculosis bacillus. That's its mechanism of action. Proof of concept that works, that it increases Tregs and B-regs and decreases pathogenic T cells without an immunosuppressive type signal in early phase studies. So I think that one would be very exciting. And then other small molecules like JAK inhibitors are going to be tested in EoE. So I think we're seeing a lot of activity and a lot of trends. We'll see, I think, based on Nurit's work, whether we can find targets in the barrier. That would be, I think, the most upstream possibility as well. So lots of possible ideas there.
Thanks so much. Yeah, we have just a couple minutes left, so I just want to ask one last question. That you ended with a vision of what EoE care might look like in 2036. Including non-invasive testing, phenotype assessment, predictive treatment selection, and non-invasive monitoring. Which of these do you think is closest to becoming part of routine care, and which remains the biggest hurdle?
I think the less invasive monitoring is here. There are places doing hundreds of unsedated transnasal endoscopies in kids each year. There are places that are really ramping up the string tests now and using that for monitoring. That's something that we're offering here at my center now for selected patients. And so I think that monitoring is here. I think we know the predictors now. And so I think the next thing for the predictors is to get them assessed in prospective trials, either prospective cohorts, some of which have been proposed already, or clinical trials. And that's more of a funding issue. But I think we need to validate the ones that we know we have now that look like they work pretty well, because that'll be the first step. And then with that, as more and more large scale clinical trials of novel therapies come through, it'll be crucial really to be assessing biomarkers and predictors in those studies. So I think those are the ones that are closest. I think we're a bit a ways from a blood test and a screening test for EoE. I think we're probably a bit away from sort of the true personalization where you like take a biopsy, send it off, and it comes back and tells you what treatment to use.
Thank you again, Dr. Dellon. With that, we'll move to closing remarks. I want to thank all three of our speakers, Dr. Jensen, Dr. Azouz, and Dr. Dellon, for excellent presentations and discussion. Thank you as well to everyone who joined us for today's IES webinar, Frontiers in Eosinophilic Esophagitis, The full webinar recording will be available on demand after the session. Your feedback is very important. Please take a moment to complete the brief post-webinar evaluation. You can use the QR code shown on the slide, and you should also look out for the post-webinar evaluation link. For those interested in joining or renewing your membership in the International Eosinophil Society, additional information is available through the IES membership link shown on the slide. Finally, we want to thank Amgen and AstraZeneca for their funding support of this webinar. And thank you again for attending. That concludes today's webinar.